All right, I have no idea if anyone's even here because yeah, software's being a little weird. So yeah, hopefully I'll give you some folks some time to show up. But the stream that I made and spent some time making lasted 1 second because I clicked a button twice, and so yeah, hopefully we'll get some folks in here. I posted a new link on Google Classroom, so if you have friends, you can let them know that that is a thing. Yeah, just completely the normal way of things. It's the only way I can say. And I think the thing takes a little bit to show who else, if people are here or not. So I don't really know if you're here. I have no way of knowing that. I can see that there's people in. Okay, there's certain people are starting to show up now. Yeah. So again, sorry about that confusion. But I haven't used the software since May, so yeah, it's a little bit still kind of wonky to me, but I'll wait a few more minutes, and not minutes, seconds even. Just kind of see if anybody else is going to show before I start. And if you have questions, you can go ahead and start putting them in the chat, and I will get to those concurrently. That's not the right word. I can't get to them all at the same time. I'll get to them in some fashion. Why does AP Power have 11 units on con? It's weird. It's weird. So yeah, I'm looking through the different sections. So if there's anything that you'd like for me to do, like questions on or not, you can let me know. But yeah, I should have gotten this started like 15 minutes ago, or no, 15 minutes prior to when I said I was going to start it, and worked out all the kinks, but that's not what happened. All right. I don't know how many people are here. It looks like just a handful, which is fine. And we'll go until I'm tired, or until you guys are finished. But I'm not seeing any questions in the chat either, and so I'm just going to work a few questions, and we'll sort of go from there. I'm going to pull these questions off of Khan Academy, just so you know, and okay. Basically, what I'll do is I'll copy paste them into OneNote and then use that as my primary vehicle, just so we don't have all the other ads and business up there. All right, so here's a question. bodies of water, such as ponds, freeze from top to bottom, forming a floating sheet of ice. This can help aquatic organisms survive during cold months, which the following statements explains why ice floats. All right, so before we go down to ABCD, we're going to just think about it in our head. All right, we talked about this. We said that the reason that ice floats is because as... yeah, I will get to that. Thank you for that question. We talked about why ice floats, and we says that as the water cools, the molecules slow down, and because certain elements of the water repel and certain elements attract, they sort of form this lattice structure, which causes them to sort of separate, right? And then because of that, they grow in mass, but they grow in volume, but stay the same mass, and so they decrease density, and it causes them to float. And so short answer is solid is less dense than liquid. The crystalline structure of solid ice makes it less dense than liquid water. B is the correct answer. Final answer. Yeet. All right, so we should go over lipids and proteins. That is okay. So yeah, I will do that instead of these questions. All right, so here's lipids. This is the same slideshows that I had at the house, and or not the house. I'm at my house at school, and so any specifics or just sort of broad brush overview. I can do broad brush overview until I see a specific question. All right, so lipids are nonpolar, right? At least some element of them is nonpolar. So as soon as you see the word lipid, think nonpolar, unless there's some kind of special lipid, which we'll look at. And the reason they're nonpolar is because there's an equal sharing of electrons, like a bond like this, right, where there's no polarity, there's no partial charges, there's no negative, positive sort of thing. And you see that with like this molecule as well, equal sharing of electrons. Lipids are things like fats, oils, waxes, steroids, and there's some special lipids like phospholipids. Lipid structure. Monomers are fatty acid, polymer, triglyceride. Typically, when you see a lipid, it's going to look something like this, right? It's going to be this long chains of carbon and hydrogens, which is significant because the main function of lipids is to store energy, and every one of these bonds has some energy in it, right? Which is going to be much more bond, or much more bonds than like a sugar or something like that. So it's going to have a better capacity to store energy. Lipids are used for other things, but that is their primary structure or primary function. And these chains are called fatty acids, and they connect to this glycerol here to make a triglyceride. And how do you connect them? Remove water, dehydration, synthesis. How do you separate them? Add water, hydrolysis. Remember that. Saturated fats are completely full of hydrogen, and because of that, they are solid at room temperature. So one of the AP classroom questions that I saw had to do with fluidity, and so a saturated fat would not increase fluidity, right? It would decrease fluidity because it causes things to be more solid, whereas unsaturated fats would increase fluidity. I think it's important to note the difference there. Unsaturated fats, because they have these double bonds, are liquid at room temperature, and that's going to factor into how they act versus saturated fat. So here's animal fat, animal store that is fat as adipose. All right, phospholipid. Let's talk a little bit about phospholipids. So first of all, you see the lipid portion of the phospholipid. You have a saturated fat and an unsaturated fat. You have the phospho portion of the phospholipid. Why is it called that? Because you have this phosphate. Phosphate has a charge, right? Charge likes polar things, and so with a phospholipid, you have a non-polar portion or a hydrophobic portion, and you have a polar portion or a hydrophilic portion. And we'll learn more about this when we get to unit 2, I believe. Yeah, 2. But because of that, in the cell, they orient themselves this way so that the non-polar portions are facing each other and the polar portions are facing out toward the outside of the cell. and in toward the inside of the cell because that's where water is. And you'll see this is called a phospholipid bilayer. This allows the cell to be only permeable to certain things, right? Because of that hydrophobic barrier or that nonpolar barrier. Steroids were like chemical messengers. Certain kinds of lipids, waxes were just talked about uses for plants, and animals have waxes as well. Energy storage. So man, this was like steroid system regulation, that sort of thing. And insulin protection. So that's just a few of the other purposes of lipids, but think energy storage. Think phospholipid. As far as like what are they made out of? They're made out of three things primarily, right? Carbon, hydrogen, oxygen, and you have these long chains of just carbon and hydrogen. The only time that you're going to see anything else is when it's a phospholipid and you're going to have phosphorus, hence the name phospholipid. But when you see a phospholipid, it's going to be so plain, right? It looks like a little weird alien or something, right? It's so plain that you're not going to have to like pick it out of a crowd. You're going to have a question about a phospholipid. What was proteins? All right, let's look at proteins. Proteins are made up of amino acids, right? Amino acids, a couple things about amino acids. First of all, proteins have carbon, oxygen, hydrogen as well, but they also have nitrogen in them. And this nitrogen is the amino group here, hence the name of the amino acid. This is what our kidneys do, and liver. So what they do is they clean this, and they clean the nitrogen out of our bodies that is made because we digest proteins. Carboxyl group is the other side of the amino acid, and the R group up here is the very variable portion of the amino acid. It's the R group that's going to cause a protein to sort of fold up in different ways, right? And we talked about this some, and then you guys, hopefully you worked the AP Classroom questions, and there's some really good questions on there as well about that. Here you see two amino acids. Notice they're oriented in such a way that if you take an OH out of one, H out of the other, you take water out, you combine them, you have a peptide bond. It's a covalent bond. It's dehydration synthesis. Question I got today from a student, and just to relay to you, remember everything's the same about amino acids except for this variable group, and the variable group is where you're going to get things like polar and non-polar, positive, negative charge, stuff like that. The rest of it is pretty straightforward, right? It is these R groups where you're going to find that variability. And so if a protein is largely nonpolar, why is that protein largely nonpolar? Because it has a lot of nonpolar amino acids. If it's mostly polar, why is it mostly polar? Because there's a lot of polar amino acids. And a protein can have sections of both, like a phospholipid, right? All right, yeah, we'll talk about that. Good question. I will answer that question. And we can answer that question here, I think is good. So why does water provide an environment for chemical reactions? So because water is polar, all right, so if you stick, well, all right, let's go up here to lipids. This is not going to dissolve in water, right? It is not going to dissolve in water because it's nonpolar. That doesn't mean that your body or like a living system can't use it. It's just not going to dissolve. Now, that's actually good if you're a steroid and your job is to be a chemical messenger and you're a lipid and you need to pass through a bunch of lipids to get to where you're going, right? If you're polar, like let's say you're a carb, which is a polar molecule, then that could be dissolved in water, right? Which is like sugar, right? If you take some water and you take some sugar and you stir it up, you get sugar water, right? And so you can dissolve that in there. Why? Because water's polar. It sort of dissolves this. Water's a universal solvent, right? And so the way that living systems use that is some things are soluble in water, which changes sort of the internal chemistry of a cell. And some things are not soluble in water, right? Like fats and other things like that. That's going to be the extent of it, I think. Nothing more than that. It was a good question. Because it kind of brings all this together. That kind of question does. Okay, levels of structure. Primary structure of a protein. It's built by amino acids, just a string of amino acids. There's a really good question in the AP Classroom questions about if you take out one of the amino acids, what does it do to the whole structure? Well, it's going to mess up the primary structure, right? Because the primary structure is obviously a string of amino acids, which is going to in turn mess up the secondary structure, which is going to in turn mess up the tertiary structure, right? And so when you change these amino acids, you change the structure, which is going to change the function of the protein. These are covalent bonds. These alpha helixes, beta pleated sheets are held together by hydrogen bonds. And then you take a bunch of helixes and sheets and you add them together and you get a tertiary structure. And the tertiary structures are held together by different sorts of bonds. Covalent bond like the disulfide bridge we talked about, but it could be lots of things. Could be hydrogen bonds, could be ionic bonds. You don't have to know all that stuff. So, and then some proteins are quaternary proteins, which are multiple tertiary structures together. We mentioned enzyme function. I'll only talk about this. Anytime you have proteins being mentioned, you're going to see enzymes. You don't need to know active site, enzyme, substrate complex, all that stuff at this point. You just need to know an enzyme's shape determines its function. That's pretty much going to help you sort through any questions about enzymes. We mentioned they make up muscles and nails and hair, kind of structural components. Immune response, lots of different lots of elements of immune response are proteins. So, good questions. I'm going to continue working on some con stuff then, if you guys don't have any specific questions. If you do, ask away. If not, I'm just going to copy paste these in there and we can look at them together. And do that. Okay. Consider the following molecule. How do disaccharides connect to polysaccharides? Dehydration synthesis. Take the water out. Add them together. Same thing. That's it. There's you learned, like we talked about monomers joining together, but if you have two and two, how do you connect them? It's the same thing. Right, it's the same idea. Based on its elemental composition, which the following molecules does this model likely represent? Okay, so what do we have? We have oxygen, we have nitrogen, we don't have any phosphorus. What does this look like? Well, you know what this looks like to me. There's a carboxyl, there's an amino, there's alpha carbon, no R group here. This is a amino acid. Which one says that? Not A. B says it. I'm not even looking at C and D. I don't even know what uridine is. It's not a thing. Glucose is a carb. Carbs don't have nitrogen. We know that it's not a nucleic acid because there's no phosphorus, right? So B is the correct answer. Moving on. Oh, this is a fun one. In the reaction depicted above, maltose is broken down into two molecules of glucose, which the following best explains this reaction. So it's a hydrolysis reaction. Add water to break, not release. Why? What do I know? Not ionic bonds. We didn't learn about that. It's not important here anyway. It's not ionic. Reverse reaction would require the addition of water to build. That's what is this? Reverse. That's a weird choice right there. Well, which involves which best explains the process of this reaction. Well, the reverse is, okay, come on. This reaction is add water to break the covalent bond. D is the correct answer. Yes, I see that question. And, okay, can we go back over the 3 to 5 on nucleic acids? Absolutely. Let's do that then. Okay. So here's a nucleic acid. How do I know? Phosphate, 5-carbon sugar, and a nitrogenous base right here. And the numbers 3 and 5 are based upon the carbons on the outside. What I mean by outside is a molecule of DNA. Let's go here. Is made up of these two strands, right? And these strands have a nitrogenous base, which you see in the middle, and these sort of spiral strands, which are made of that sugar phosphate backbone, right? You've heard me talk about the sugar phosphate backbone, which is what you see here and here, right? This part in the middle, these T and A and C and G are the nitrogenous bases, but this phosphate, sugar, phosphate, sugar, sugar, phosphate, sugar, phosphate, that is the backbone. And so that is what we see here. The base is just the word base. And so these carbons form that outer edge. And the 3 carbons, 1 is always next to the base, then 2, and then 3. 4 is unimportant because it's in the middle, just like the middle child. Just teasing. Just teasing. And then 5 is up here by the phosphate, right? And so that direction matters because this molecule, if you turn it upside down, then the 3 is up, right? And the 5 is on the bottom. And so one of the mistakes that students will often do is they'll say, well, which direction does it go? Well, it depends on what you're doing with it, right? So that is the important thing. So if you come down here and look at this molecule, you may think, well, is 5 always on top and 3 always? I heard someone say that today. No, because over here it's not the case, right? But a molecule is typically read when DNA is being read, right? It is always read from 3' to 5', which is like you always read left to right, top to bottom, right? And so why does that matter? Well, here, if we could go back down to this, if we wanted to read this molecule on the inside, I don't know why these arrows are there. If we wanted to read this molecule on the left, or this strand on the left, we'd have to go from 3 to 5. We'd go up. If we wanted to do the row on the right, we'd go from 3 to 5, or we'd go down. New bases are added to the 3' end, right? And so as this is a strand of RNA being made, not important on how this strand is made in this unit, but this strand of RNA is being made. As new bases are being added to this RNA, they're going to be added to the 3' end, right? That's all that is. The shock of this is that these carbons have names and these 3's and these 5's matter, right? How are you going to see a question like this? It would be something to the effect of the direction that DNA is read, where new bases are being added. So if it's like DNA replication, or something like this, where's new bases being added, you don't have to know those processes. You just have to know, where am I putting a new base? Now, hopefully you've seen enough questions by this point to realize that if the question has replication in it, they're going to add a bunch of weird crap that you don't have to know, right? But just focus on the things that you know, pick out all the weeds, and find the thing that you know to be true, and then go find the answer. Because all of those weeds are that's all distractions. It's all the hardest part about this class is weeding through the distractions, not the content. You guys are good at studying content. It is learning how not to get distracted so that you can actually apply the content that you know. Because I don't doubt that you know the content. It's that skill of dealing with hard questions that's hard. Hopefully that was helpful. If I didn't answer your question, let me know. But we'll keep going through these questions. I like these questions. All right, so 1950s, the scientists Alfred Hershey and Martha Chase, we talked about them, remember them, carried out an experiment using bacteriophages, which are viruses that infect bacteria. During the experiment, scientists allowed bacteriophages, been labeled radioactive element, to infect bacterial cells. After carrying out their experiment, the scientists concluded that bacteriophages inject DNA, a type of nucleic acids, but not protein, into the host bacterial cells. We know that, right? We know that from our unit last year. To be able to form this conclusion, which the following radioactive elements did Hershey and Chase most likely find in the infected bacterial cells? Okay, so how did they come to the conclusion that it was DNA and not proteins? Well, what is found in DNA that is not found in proteins? It's only one thing. It's phosphorus. It's not carbon, hydrogen. Sulfur's the distractor here, right? Because you know that carbon and hydrogen is not, or is found in both. Sulfur's like it's going to make you think, or it's going to make you think about it backwards, right? Because sulfur's found in proteins but not in DNA. And so the answer is A, phosphorus. Here's a fun question. Air temperature ranges of several Southern California cities in August are shown. There they are. Which of the following best explains why coastal cities like San Diego often have smaller air temperature ranges than inland cities? Okay, it's a great question. So before we go down and look, I don't even know what these other cities are. They're inland cities, right? We know that because San Diego's coastal. And let's go up in the data table and confirm what the question is asking. So San Diego's right on the coast. Notice the other ones are more inland. And it is also telling us that there's not as much of a temperature change, right? Yeah, and we see that. We see that the average daily temperature is 25 and the nightly temperature is 22. Very small swing, as opposed to 29, 17, and 42 and 25. Why is it the case? So it's something about being on the coast, right? Well, what does water do with temperature? We know that temperature's cool at night. Why does it cool at night? Because it's dark. The sun's not shining on that side of the earth. And so we also talked about how when that occurs, all the energy that's been trapped in water all day releases back out into the atmosphere. Where's that energy going to go? It's going to go to the surrounding land, which San Diego happens to be nearby. And so it's going to be less likely to cool down, right? It's going to have a more steady temperature. Let's go find that, ABCD. High capacity allows the ocean to moderate the temperature of nearby air. I'm done looking. We'll go look at why the other ones are wrong, but that's it. The cohesive tendency of water causes high surface tension. No. Because water is less. No. Adhesive properties of water allow water molecules to stick together, preventing temperature changes. This is probably a distractor, maybe, because it's got the word temperature in there, and you're like, I don't know, right? So that may be good, but I like A. Hard work paid off. Congrats, going to the next level. Woot. Man, keep this up. I don't need to see a summary, but I don't have a choice. It said you can skip, but then I tried to skip and I couldn't skip. All right. So that was the quiz. We're not going to take that quiz again. We're going to do quiz 2. Time for a quiz. 5 questions. Let's go. focus. All right, here we go. In order to develop antibiotics, scientists need to understand how bacteria grow, divide, and spread during infection. Essential part of bacterial growth is RNA synthesis, because RNA can be synthesized before protein can be assembled. If RNA synthesis block proteins, or bacteria will not be able to reproduce. Which of the following questions best love these? Okay, so this is a great example of a type of question you'll see in the exam. You'll probably see 3 or 4 of these on the exam. Which of the following questions will best direct an investigation on how an antibiotic that inhibits RNA synthesis in bacteria can be developed? And so it's asking you to come up with an appropriate question, knowing what you know about RNA, right? And RNA synthesis, we don't know anything about RNA synthesis. We just know something about RNA. What would block the production of proteins? We want to block RNA, right? And so let's look. Let's find the right question. Which proteins are used to undergo cell division through binary fission? No. Doesn't matter, right? Doesn't matter. Which chemical compounds block the 3' end of a growing RNA strand? Okay, we like that one, right? Because we learned that new bases are added to the RNA strand as the RNA is being made. But we're going to hang on to that one. I think that one's probably right, but I want to be sure. Which chemical compounds add a hydroxyl group? No. Which chemical blocks the 5' end? Okay, good. So yeah. So the question is, where are new bases added, right? Are they added to the 5' end or the 3' end? And unless I just got it completely backwards, which I've been known to do. B is the correct answer. Good. All right. Human immunodeficiency virus, HIV, is classified as an RNA virus because its genome or genetic material consists of single-stranded RNA, which is the following best claims. Best supported by the information above. So this is a claims, right? Making a claim about information that we know. Nucleotides of HIV form an anti-parallel. No, because it's a single strand, right? What? Structure of HIV forms. No. Uracil nucleotides are present. Yep, we know that to be true because uracil is found in RNA. This is one of those questions, and I know some of you, because I had you in class last year, this is one of those questions that is so easy that some of you are going to trip over it, and you're going to be like, well, Mr. Chipman said that RNA can double up on itself, so what? It might be this. But D is so right that nothing else can, even if you like dream up stories about A and B and C, D is still going to be wondering, me like, what? I'm right. And so just don't do that. Don't create stories. We'll do a couple more. It's getting late. We'll finish this quiz. In order to develop antibiotics, did we answer this question? It looks like a new one, though. It's similar. Which of the following best directs an investigation on how to develop antibiotics that inhibit protein synthesis? Yeah, it's a similar kind of question, but different. How can the formation of covalent bonds between amino acids by understanding how bacteria synthesize proteins? A looks good, right? We're not worried about a hydroxyl end because there's not one. There's a carboxyl end. Phosphate end, nope, not in proteins. Formation of hydrogen bonds, nope, it is A. Another one, that one actually wasn't hard at all. Moving on, let's see. Which the following statement accurately describes the difference between RNA and DNA. DNA has 3' hydroxyl, 5' phosphate ends, but RNA has DNA has 3' hydroxyl and 5' phosphate ends, but RNA has 3. They're the same. I'm not even going to try to parse through that. That's not a difference between them. In DNA synthesis, nucleotides are added to the thread. No. DNA is usually double-stranded. RNA is usually single-stranded. See? So, like, another one, great example. I told you the differences between DNA and RNA, right? And they were like single, double, uracilthymine, deoxyribose, ribose, functional differences like stability and what their function is. Don't try to even like spend time parsing through the 3 and 5 garbage, right? It's just as simple as this. That's it. Well, what? No, they're not close to right. They're just wrong. Oh, this is a big question. Just noticed it's really big. Lots of words. That's why they zoom out. Okay. In the early 1950s, a team of researchers led by Erwin Chargaff conducted a series of experiments related to deciphering the chemical composition of DNA. In one experiment, Chargaff's team examined samples of DNA from different sea urchins. Sea urchins, eh, Chargaff? I wonder why. The result of the experiments were summarized below. I think I saw this one on somebody's Chromebook today. And so you have their sea urchin species here, molar ratio, fancy for like just, even if this was gone, even if this said like reindeer ratio, it wouldn't matter what it says, right? It says adenine to guanine. These are like ratios, right? And so what is the ratio of adenine to guanine? What is the ratio of thymine to cytosine? And so forth. It's basically comparing them, right? Ratio to purines to pyrimidines. So let's look. First of all, which of the following results from the experiment is most consistent with base pairing rules? First of all, what are our base pairing rules? Is that A pairs to T and C pairs to G. So when you compare adenine to thymine, it's 1 to 1, right? When you compare guanine to cytosine, it's 1 to 1. All this business up here, meh, unimportant. What we want to know is that A and T are the same and C and G are the same. Or purines are the same as pyrimidines. Percentage of adenine nucleotides is roughly equal to the percentage of thymine nucleotides across all species. Boop, boop, boop. A is the correct answer. Not even reading the other ones because you know they're wrong. And I get the happy chimes. The happy chimes. Not seeing any chat questions, which is great. But it's 9:30. It's probably time to go to bed. And hopefully this was helpful. If you still have questions, I'm in my room early. The doors open to the school at 7:30. Come see me. I'll be in my classroom, and we can chat about AP Bio related stuff. But your best bet is, besides just getting in bed and getting some good sleep, is doing your AP classroom questions and doing these con questions, because chances are you've already stared at your notebook for long enough, and so now you need to go and see if you actually do know these things and apply the information that you know to these types of questions. The more, I mean, it's like the difference in like, all right, so if you play baseball or softball, you'll get this. Or if you play an instrument, you'll get it. You can like read about batting, but actually facing a pitcher is going to help you get better, right? Or you can read about playing an instrument, but actually playing it is going to help you get better. And so you need to think about these questions as actually doing it, right? And you need to think about your notes as reading about it. Sure, that's important. But going out and actually using the information that you have is going to be the most important part of getting better and better at this class. Hopefully that was helpful. See you guys in the morning.